DNA replication is an important process in the cell where a DNA strand is converted into two identical DNA strands. Here we have our DNA strand. The Twizzlers represent the sugar-phosphate backbone of the DNA, and the sprees represent each nitrogen base. The top strand is the leading strand. It is read in the 3' to 5' direction. When replication occurs, the new daughter strand is formed 5' to 3'.
Topoisomerase II comes along and relieves the stress of unwinding of the strand. Helicase breaks the hydrogen bonds between complementary nitrogen bases in the DNA and separates the strands like a zipper. All the while, single-stranded binding protein stabilizes the strands and keeps them from joining back together.
RNA primase sets an RNA primer as a starting place for DNA polymerase.
Once the primer is in place, DNA polymerase comes in and extends the primer with a new sugar-phosphate backbone and the appropriate nitrogen bases. DNA polymerase comes in and removes the primer and replaces it with DNA.
Here, replication of the leading strand is shown. This replication is continuous because the parental leaving strand is 3' to 5'. Therefore, DNA polymerase can create the daughter strand 5' to 3'.
Now we are showing the replication of the lagging strand. Because this parental strand is 5' to 3' and the daughter strand must also be created 5' to 3', the process is discontinuous. To accommodate for this, multiple RNA primers are laid down along the parental strand. DNA polymerase extends these primers with a new backbone and nitrogen bases, but does so in fragments called Okazaki fragments.
DNA polymerase removes the RNA primers. After that, DNA ligase comes in and joins the Okazaki fragments like a sticky caramel candy, forming a complete continuous strand.
As the process continues, the two parental strands are transformed into two identical structures of DNA.